we're going to stay pretty much on this same theme of looking at life within serpentinites here, but from somewhat more of a theoretical view than what Alexis talked about just recently, in the prior talk. But I have mercy on you this afternoon. I only have one data slide in this talk. So, and this is work that I've been working on for several years, and have a number of collaborators that have worked on helped me understand different aspects of this listed up here. So we are looking at this serpentinization reaction. I think most of you by now are pretty familiar with this idea that mantle peridotite rocks containing olivine and pyroxene react with water at temperatures below about 350 to undergo this serpentinization reaction. And the reason this is of interest to astrobiology is because one of the byproducts of this reaction is the production of hydrogen, which can then go on and feed microbial populations in these serpentinizing environments. And over the last couple of decades, several of these kind of environments have been the focus of different microbiology studies. Most of these have focused on places like hydrothermal vents on the ocean floor, like places like Lost City and Alkaline Springs, where these fluids are coming up and being discharged at the surface, where the interaction with surface oxidants presents a favorable habitat for biology. What I'm really going to be looking more at today is the subsurface part of this systems, where you have either seawater or groundwater penetrating into these rocks, reacting with the olivine and pyroxene, and undergoing this serpentinization reaction. And what I'm looking at here is the capacity for these rocks to support life in these subsurface environments, so below these springs and hydrothermal vents. What can live there, and how much biomass it can support? So I'm looking at a couple of questions. Can autotrophic communities exist within the subsurface of these serpentinizing systems? And if so, how much subsurface microbial life can be supported in those kinds of environments? So I'm looking at this from a kind of an energy balance perspective, where in some system there's an energy supply, and the organisms that live in there have some sort of demand of energy in order to run their metabolism. So different environments can have different degrees of energy supply, and in different environments organisms can have different energy requirements in order to exist. And if the energy supply far exists the demand, then there can be growth of organisms. But if it's not sufficient to support microbiology, then life is not possible. And then there's an intermediate level where organisms can exist there but maybe not actively grow. And kind of the hydrothermal vents and alkaline springs environments are probably some up here where there's a lot of energy supply to the environment, but most sort of subsurface environments probably exist somewhere along this interface where there's enough energy to stay there and live there and survive, but not a lot of energy for growth. So I'm going to first look at the energy supply in these serpentinizing systems and ask the question, how much energy is available for hydrogen-based lithoautotrophy in these subsurface environments? So we have this reaction where olivine and pyroxene is creating this serpentine mineral assemblage and hydrogen. And one way to get at how much hydrogen is potentially being produced in these environments is to look at reaction rates. And here's my one data slide. And what I've done here is I've compiled all the experimentally derived data for hydrogen production during experimental serpentinization, so experiments that started out with olivine or olivine plus pyroxene and measured the amount of hydrogen coming off this thing. So I'm plotting these up as a function of temperature. This is hydrogen production rate in the units of nanomoles per gram per day. And this is something that is really people have only started to experiment this, to investigate this experimentally in the last 15 or so years, so there's not a lot of data to constrain this. And you'll notice right away that most of the data that's available is present at temperatures that are higher at about 230 degrees. Another thing you'll note is that, at any one particular temperature, there's about a 4 order of magnitude distribution or variation in the rates that have been measured in these laboratory experiments. These are due to factors such as rock composition, pH, and we're just really beginning to understand what causes that kind of variation and understand why different kind of rocks can produce different amounts of hydrogen in different situations. One thing you'll notice too, that there's pretty much a scarcity of data at lower temperatures, where we were looking at these potentially habitable shallow subsurface environments. And all of these data that's shown here with arrows on it, those are experiments where there was too little hydrogen presented to measure it on laboratory timescales. So pretty much at temperatures below about 175 degrees, the reactant is just going too slow to be able to measure hydrogen production from serpentinization on laboratory timescales. So that's already giving us a clue that in these shallow, low-temperature environments, the hydrogen rate of hydrogen delivery by the serpentinization reactant is going to be pretty slow. But if we go down here, if we take just bracket these higher temperature data with a very simple preliminary and probably not very good model for first order reaction kinetics with Arrhenius temperature dependencies, we can just draw in some provisional lines here about what we think might be the hydrogen production rate in low temperature environments. And I should say too that these are all measured on laboratory conditions where you have powdered minerals and very favorable conditions for the reaction. And in a natural system, we would probably expect things to be going actually slower than these. But if we just take this data that we have and extrapolate it down temperature, if we go down here in like the 50 to 30 degree temperature range where we think things might be living in subsurface environments, we can extrapolate to something like a rate of 10 to the minus 18th or 10 to the minus 14th moles of hydrogen being produced per gram per day in these shallow subsurface environments. So we get something like that. That translates to something like 10 to the 6th to 10 to the 10th molecules per hydrogen per gram of rock per day. And just for perspective on that, some cell-specific rates for sulfate reduction in marine sediments, that requires about 2,500 molecules per day per cell in order to maintain that. For aerobic heterotrophs, they consume about something like 10 to the 5th molecules of oxygen per day. So right away, we're getting to see that this is probably going to be only enough to support a relatively small population. We can look at this energetically also and say, well, if we have a reaction like sulfate reduction that's using this hydrogen that yields about 10 kilojoules per mole of hydrogen, this would equate to an energetic power supply of something on the order of 10 to the 15th to the 10 to the 19th watts per gram of rock. So that's our power supply. What about the energy demand? Well, this is something that's really not known very well at all about how much energy is the minimum amount of an organism can survive on. One study recently that has been looked at this is this paper by Doug Leroux and Jan Ammann, where they looked at microbial populations in submarine sediments in the South Pacific Gyre and compared it with various models of the minimal energy supply. And they came up with an estimate of the basal maintenance energy requirement for a cell of something like 2 to the minus 10 to the 19th watts per cell, or 200 zeptowatts of energy. So we can compare this with our serpentinization supply of energy, and it looks like there's enough energy being supplied to support less than only a few hundred cells per centimeter cubed. And this may be an optimistic number based on these laboratory experiments, and the actual hydrogen generation rate in natural rocks may be somewhat lower than that. But I'm not sure that kind of maintenance energy requirement applies so much to these high pH serpentinizing environments where the high pH may impose additional energetic costs. For example, I took this from some work by Holler that's in preparation, some preliminary calculations of what it costs to maintain your energy, the pH across the cellular membrane if you're consuming something like bicarbonate or sulfate with hydrogen. And as you go up in pH, the cost of maintaining your cellular membrane potential increases substantially. So we may be looking at additional energetic costs just to live in those kind of environments more than what we see in those other estimates I showed. So it may cost more. But in any case, it looks like very small populations can be maintained in these subsurface serpentinizing environments based on hydrogen. And that seems to be consistent with some data that are beginning to come in looking at these environments. And for example, this is some data that came out last year from drill holes drilled into the Atlantic Massif, where the Lost City system is hosted here. So looking down these drill holes, what they find is cell populations in those rocks on the order of 10 to 100 cells per centimeter cubed, so very small populations, and that may be all that this hydrogen-producing serpentinization reaction may be able to produce. Another environment where astrobiologists are interested in this in the solar system is Mars, and because we see serpentinites on Mars and we have a surface that seems relatively inhospitable, there's a lot of people that are proposing that we need to go look at subsurface environments, and serpentinites are certainly a good target for that kind of thing. But these extremely low biomass populations may indicate that these impose really serious challenges for detection of life within serpentinizing rocks. So if you're trying to find a rock on Mars that only is supporting a couple of hundred or less cells per centimeter of rock, it may be tough finding those biomarkers. What may be more favorable is to go to look for places like these alkaline springs or other areas where fluid flow is focusing these energy sources into a more confined environment, and may be better targets. Those places may be better targets to go and look for evidence of life in Mars and other planetary bodies beyond Earth. Anyway, that's all I have today, and happy to answer any questions.
Thanks, Tom. Yep.
You showed a diagram of the rate of H2 generation versus temperature.
Yes.
And the highest rate was around 250 degrees Celsius. However, you did not, I did not see the pH. Yes, it's on the...
Well, that's one of the pH is probably one of the variables that accounts for this range in hydrogen generation rates. And actually, these ones in red, those are all experiments that have a room temperature pH greater than 10. Everything else has a room temperature pH below S. So in some cases, you see faster hydrogen generation at higher pH. Some cases, you don't. And that's something is subject of ongoing investigations of why that happens.
Yes, I think pH is very important for H2 generation, because if you look at a diagram of the potential, electric potential versus pH, you can realize that at high alkaline pH, very high, 9.5 to 14, and at high temperature, 300 to 350 degrees Celsius, and also 10 to 25 megapascal, the ferrous ion is transformed into ferric ion with emission of H2. So you approach the temperature, but the pressure is also very important.
Yes, well, yeah, and these experiments are almost all performed at about 350 to 500 bars.
Yes.
It's high.
So this is the correct pressure because 300 bars is 30 megapascal.
Yeah, I would
so
agree. I would agree that high pH and these temperatures, there is a lot of thermodynamic potential to create higher amounts of hydrogen, but these rates are pretty much all controlled by kinetic factors. So you have to add that into the thinking about what is thermodynamically favorable.
Yes, but I just wanted to show that in this specific domain of water, which is called high subcritical, and also where the silica dissolves much better than at lower temperature and higher temperature, all these compounds will dissolve and form H2 and SiO2. Maybe if you still have time, in 5 minutes I can show you my poster about that, and I shall send you my articles.
Yeah, please do. I'm glad to talk about that more.
Because I think we should probably move. Oh, a quick question?
Just a quick question, yes. I mean, that's great work. One thing I'd like to just question is, when we talk about Mars, applications for Mars, you use in situ as the argument. That's why you're looking at the lower temperatures. Now, maybe when you think about biomass estimations for Mars, we should not think all the time about in situ. We have gradients. So if you go, think about Mars water percolation, you can have actually water mostly in contact with much warmer regions where you have much larger production, in the order of below 100 C. So you can produce much more and bring it into the regions where there's more maybe life. So which means you will be you'd have production rates which are orders of magnitude larger than the lower temperature ones. So I would be careful using the in situ low temperature as a prediction for biomass, because it might be quite misleading.
Yes, and I absolutely agree. And I think a lot of these systems, for example, the one in Oman, those kinds of environments, I think they are largely driven by higher long residence time fluid at probably higher temperatures that are carrying their hydrogen and methane load up towards the surface where it begins to be. So it's kind of a focusing mechanism there. Yes. So that of course changes the whole picture. Yes. If that's right.
Thanks, Tom.
Thanks.
Yep, appreciate it.